So all bodies have bones, and all bones are full of bone marrow. And it's from the bone marrow that all of our circulating blood cells develop. Now, the bone marrow isn't made up of just a random clumping of cells, but it's actually a highly organised, highly vascularised system of niches or microenvironments. And these microenvironments act a bit like a childcare for developing blood cells, where these precursors are nurtured and directed to develop into young adulthood, where they take on their different shapes and sizes before they move out into the periphery to do whatever it is they're best suited to do. Now, at the same time, the bone marrow acts a bit like an early age retirement home where mature cell types return to the bone marrow to be maintained until they're needed again or until they die. So the bone marrow is vital for maintaining and developing cells that we need to fight infection. Now, a whole range of pathogens can directly infect the bone marrow, and they can do this as a clever immune evasion strategy whereby they kill off the developing blood cells as well as the mature blood cells, and so we have less cells that are able to fight an infection, and so the pathogen can become more virulent and more persistent. So we have dendritic cells as well, DCs. These are antigen-presenting cells. They're vital for initiating and promoting immune responses. They're found throughout the body, but wherever you find them, you find different kinds of dendritic cells that are better at launching different kinds of immune responses. Now, we know that there are dendritic cells, DCs, in the bone marrow, but no one's ever had a really good look to find out which ones are there and what they're capable of doing. So my PhD thesis can be basically broken down into 2 points. Firstly, to establish exactly what kinds of dendritic cells are in the bone marrow, and secondly, to see how these bone marrow DCs might react during a viral infection setting of the bone marrow. What we've found so far is that there are at least 3 different kinds of dendritic cells within the bone marrow, each with their unique ability to promote immune responses. We've also identified an immediate precursor for each of these 3 dendritic cell subsets. And what we found is that while these DCs might be really important for promoting immune responses, it also appears that these DCs play a vital role in maintaining cellular homeostasis within the bone marrow microenvironments themselves. Thank you.